Just sir,
Hello.
please for 1 minute and then start.
Okay. उनके पास बहुत सारे आए होंगे ना, तो एक एक करके कर रहे होंगे.
Hello, sir, we can start now.
Okay. So I start the meeting. Now our speakers has already joined. Good afternoon, Dr. Hanuman Singh Rathore sir, and all teachers, research scholars, students, and other participants. I, Dr. Dhanse Devangan, welcome you all in the fourth day of 1 week National E-Workshop jointly organized by Department of Chemistry, Geology and Botany, Government Lahari PG College, Chirmiri, Korea, Chhattisgarh. It's a great pleasure for me to introduce our today's dynamic speaker, Dr. Hanuman Singh Rathore sir, who is going to deliver a talk about biotechnology research in 21st century. I really thank to sir for giving our valuable time and being here with us in our busy schedule. So before inviting to sir for lecture, I would like to give a brief introduction of him. Please allow me, sir. Sir is presently working as an assistant professor and in charge technology, University of Nagaland, Dimapur campus. Sir is also approved PhD supervisor. Sir has completely successfully submitted a research grant under DBT Government of India as a principal investigator. The research interest of Sir is biopharmaceuticals and tissue engineering. Sir has also published number of research publications in various reputed journals. So with this word, I welcome again Dr. Hanuman Singh Rathore sir for today's lecture. Welcome, sir.
Thank you, Dhansar sir. Very good afternoon to all. My voice is clear, na?
Yes, sir. Clear.
Clear, right? First of all, सर ने कहा था कि मुझे कि लैंग्वेज दोनों रखनी है, तो मैं
जी सर.
हिंदी और इंग्लिश दोनों यूज करूंगा. ठीक है. मैक्सिमम तो मैं इंग्लिश ही यूज करूंगा क्योंकि जो टर्म्स है वो सभी इंग्लिश में ही रहेंगे. और अगर किसी को भी कोई डाउट हो या कोई डिस्कशन करना हो तो वो मुझे इंटरप्ट कर सकते हैं. ठीक है. फर्स्ट ऑफ ऑल मैं थैंक्स कहना चाहूंगा प्रिंसिपल डॉक्टर लहरी PG कॉलेज and also organizers and Dr. Dhansa Devangan also to giving me this opportunity. So my PPT is visible, yeah?
Yes, sir. Visible.
Yeah. So I will not see the chat here. I will directly go in the PPT. So I may not able to see the things. So I'm in the PPT now. Okay. So suppose if any change will be there, you or if it is disconnect something in between the lecture and all, please inform me over the phone. Okay. Dhansa sir. Okay. So good afternoon everyone. So today मैं प्रेजेंट करना चाहूंगा बायोटेक्नोलॉजी रिसर्च इन 21st सेंचुरी. So first of all, what is the biotechnology actually? So if you can see in the diagram, it is shows as like a biotechnology tree. So the term biotechnology is your it is the basically your fundamental principle which is applied by using the biological agent for good and service. That is the main aim of this biotechnology. So you can see here in this biotechnology tree, in the bottom side, you can see the different streams. These streams is generally your depend on different areas, which is generally we are studying in the biotechnology, like here human, animal and plant physiology. So human and animal plant physiology, how it is structures, what are the functions, what are the different organs, that things we are studying. So this all this your another is like a molecular and cell biology. So I will brief some few points in the each point, because I think it is the mix of audience of your botany, zoology also and chemistry also, and other streams if it is available. So in the molecular and cell biology, generally we'll see in the molecule level.
Hello, sorry to
Yes.
go to slide show option.
Is it coming slideshow now? Is it okay?
Not yet, sir. No.
Actually, when I do the slideshow here, just a minute, okay? I will open a PDF, so it will be much better. Let me, I stop this PPT now. I'll make the in PDF. So let me check, okay?
Sir, please carry on. You can do as it.
Let me try it. So it will be. So is the PDF is visible properly now?
Yes, sir, it's visible.
Now this is okay, I think.
Okay, sir.
Just okay. Let me check the slides here also here. Is it, sir, Dhansai sir, is it visible now? Okay.
Yes, sir, visible.
Yeah. So now it is okay, I think so. I hope that others are can see easily.
Okay.
Okay. So here we're talking about the fundamental principle which is involving human, animal, plant physiology, then molecular and cell biology. So in molecules and cell biology, generally we will see all the structures of your cells, and in the molecular level of your DNA, RNA, or proteins, all the building blocks. There are mainly here we will see the engineering process also. Then another is like immunology. So the immunology, it's quite familiar. It's like a response of our body against defense systems. With the your others from the environment, if we pathogens or anything enter in us, the body how it is responding against it. So that we'll deal in the immunology. Then microbiology, specifically your all kind of the microbes which is in the micron size. So it's a bacteria, virus, fungi and other things. Biochemistry is the specifically your building blocks, molecules like a carbohydrate, your proteins, fats, lipids, that thing. Genetics is the your genetically structure of your DNA specifically, and it is the transfer of the your hereditary genes to information from generation to generation. Then chemical engineering, that is also involved. So in this, if you are talking about the biotechnology, it is like a interdisciplinary term. It also required your physics and mathematical principles, as well as a chemistry in a part of. So if you see in a broad term, all this in the biotechnology, you need to study the biology, mathematics, your physics, as well as the chemistry. So it's all this principle is involved in this. So when we see in the tree part, these are your root part, which is important. Then your fundamental principle of that, the bioprocess technology. So bioprocess technology is like a producing some goods from the using your microbes or production of some enzymes. It is like a higher scale of. You can see that in the market products like your wines, beer, dairy products. These are your bioprocess technology products. And above, if we go in vitro cell cultivation. So in vitro cell cultivation, if you are using the term in vitro, means it is the outside of the body. Generally, its term used as use in the test tube. So it's not the exact term. So it's just we'll see that it is the outside from the living cells. So we are growing in the laboratory the cells. Then your protein engineering and immunochemistry and genetic engineering. So this genetic engineering is nothing but we are changing in the genetic materials. How it is, I will explain you after this next slide how the things will be. So this all the fundamentals and engineering principle, whatever is here, they have been used in the different applications. It will generally cover your all the sectors, specifically your energy and environment management. If your pollution is more, it can be used to reduce as a bioremediations, plant agriculture, crop improvements. So the more stables, or you can say drought resistance plants, can be make it or modify it by using your biotechnology tools. Animal agriculture, your a quality of the animal can be improved. Diagnostics of the different disease. Then your fermentation technology. This fermentation technology is by using your specific microbes. Not only the microbes, it can be used any cells. Either it will be plant cells, animal cells, or microbes. But generally, we are preferring much more microbes because their production time is less. But sometimes we are using some others also, animal cells, because of their production. Because ultimately our purpose is generally used for in human. So if the product is useful in human, we use generally the eukaryotic cells. Because of the product which will come up, it need your post-translational modification. Then your food innovation and food processing, that the food dairy products, milk products, this all is used as your biotechnology tools. Healthcare and pharmaceuticals. So these are also important sector in the health sector. Whatever research goes on nowadays, it's directly or indirectly related with your biotechnology. Okay. So up to now, anyone having any doubts, or they want to ask something on this regard?
सर बच्चों के डाउट्स आ रहे हैं जिसको हम लोग नोट करते जा रहे हैं.
ठीक है, तो मैं लास्ट में लूंगा.
हाँ यूट्यूब में आ रहे हैं.
Sorry. So you can see here in the diagram, in this diagram you can see the cells. So this is nothing but your central dogma. if you see the central dogma, you're generally the DNA. You can see here deoxyribose nucleic acid sequence. This. So if we see that in 20th century, discovery, you're the structured DNA, which was discovered in 1953 by Watson and Crick. So once it has been searched, this DNA, it has been solved many questions. And after discovery of the structure of this DNA, only the further research was revolutionized in the biological sector. Not only in the biological sector. If you see the Nobel laureates.
Sorry, sir. Again, I am interrupting you. Please go to next slide.
Oh, just a bit. I think I'm using the in PDF, I think, or just
Okay,
a minute.
please move, sir. Yes.
Okay, still it is. Oh, I'm sorry. This is your PowerPoint, which is, I think. Is it fine?
Yes, sir. Now
Yes,
it's
sir.
okay.
Okay. मैं तेरी मेथड देख रहा हूँ इसमें क्या बताया गया है वो पूरा पेपर निकाल दूँ मैं
So in this cell, in 1953, your Watson and Crick has given the structure of your DNA. So after this discovery of the DNA, your many thing has been changed. Not only in the biological sector, if you see the Nobel laureates, they were working in the physics and chemistry also. Okay, so this all has been changed like a revolutionized, like anything. Means if you see that whatever in 21st century we are doing is based on the structure analysis of your DNA. I will come to in the 21st century how it will be. First see the basic thing. So in this, if you see, this is your central dogma. So generally your DNA to DNA is nothing but your replication. Means from the one DNA to DNA will be replication process. After DNA, it will be transcribed to the RNA. That RNA is for further processing and getting translated as a protein. So here in this process, this is the process how it takes place. Now, this was the starting in all the cells. So now I need to ask one thing. What is the drug? Generally, what is the drug? Anyone can just inform me.
Sir, drug is a chemical compound which is used for treatment of any disease.
Okay. So now here, you are correct, Sadhan sir. What
Okay.
I feel in this, there is any chemicals which will give a physiological change. So I was just adding in that if it is change the physiology of any structures or any of the cells or anyone, that is called as a drug. Okay. So for example, if we are using any drug, it will generally block or their mechanism of actions are different. It may be in the change in the DNA structure. It will break the cells of the bacteria or other organism, or it will be blocking your translation part, or it will be affecting some other signaling molecules after upregulation or downregulation of any translation or your gene regulations. So that is your main purpose of the drug to act in any of the processes. So here in the next slide, we have in this, if you see here, the DNA is there. It is generally, we are using the term genomics. Omics is actually your like a collective term for all that. So here the genome is what actually? Genome is the contain your all the genetic material of the cell. All the genetic material of the cell is the genome. It may be depending on the cell. If it is the prokaryotic, it having the genetic material present in the cytosol, and there is some other genetic material like a plasmid also. Okay, and if you are talking about the eukaryotic, in this your mitochondrial DNA, then your nucleus DNA, and if you are talking about the plant cell, it is a chloroplast contain your genetic materials. So all the DNA material which is present in the cell we used to call as a genome. So genomics is the your all that organisms contain all the collection of the your DNA materials. So here in here DNA, then this mRNA, another term is like a transcriptomics. This is the your all the mRNA material which is we are studying in the organism. Then your comes as a picture of your proteomics. This proteomics is the your translation. So generally what will be happen? Your mRNA will take the information and then it will be coding based on the DNA triplet code. It will code for the particular specific amino acid. That amino acid will form as a polypeptide chain and make as a protein. This protein having some specific work. If it is the enzyme, because enzymes are the proteinous in nature, or any other activities. So this is the proteomics. Now the another terms are metabolomics. This metabolomics is the your all the metabolites which is present in the cell, that is the your metabolomics. So here if you are talking about in the places, we can block in some places for our purposes. Now if you are talking about the as this is the research methodology topic of your workshop, so here I will talk about the research point of view. What is it? Any research which is involved, there is a two thing is very important, the word why and how. Why means क्यों हम किसी रिसर्च को कर रहे हैं. तो कोई भी साइंटिस्ट, रिसर्च स्कॉलर, स्टूडेंट, कोई भी अगर कोई भी काम कर रहा है रिसर्च फील्ड में, तो उनको सबसे पहले ये सोचना चाहिए कि वो क्यों कर रहे हैं, उसका पर्पस क्या है. तो यहां पर साइंटिफिक कम्युनिटी के ऊपर ये दायित्व होता है कि उनके ऊपर सोसाइटी के लिए वो अपना योगदान कैसे दे सके. सोसाइटी के लिए योगदान मतलब अब वो रिसर्च फील्ड में है, तो अगर सोसाइटी की कुछ प्रॉब्लम्स है, एग्जाम्पल अगर पॉल्यूशन ज्यादा है तो उसे कैसे सॉल्व किया जाए या उसे कैसे कम किया जा सके. पॉल्यूशन में आपका सॉलिड वेस्ट है या वाटर ट्रीटमेंट है, तो उसमें अपना इनपुट कैसे दिया जा सकता है. तो यहां पे आता है व्हाई. A researcher should think why I am doing this. What is the requirement? Okay, let me give that. Now, last year we have gone through the COVID period. So कोरोना काल में ये डिसीज स्प्रेड हुआ. So जितने साइंटिफिक कम्युनिटी थे सभी ने इसकी रिसर्च स्टार्ट की. तो कोरोना इज द वायरस. तो कोविड-19 वायरस का सीक्वेंसिंग किया गया कि उसके स्ट्रक्चर क्या है, और उसके बाद उसका स्ट्रक्चर जानने के बाद उसमें टारगेट बनाया गया कि कहां पर इसे टारगेट किया जा सके. now I will come to in 21st century. Bioinformatics. What bioinformatics? Bioinformatics involve the use of your computer software tools to solve the biological problems. So if you see the wheel in this, here in this wheel, if you see this all is the your system biology, it start from the biomolecules, DNA, RNA, protein, signaling network, cells, your organism, clinician, environment. So now, if you see any of the, either it is agriculture products, agriculture related cells, plant cells, animal cell, or microbes, this all having a cells contain your DNA. So this DNA is the important to know their sequence. So here NGS is the your a 21st century revolutionary work. This is what is this NGS? NGS is your new generation sequencing. Once the DNA has been discovered, that DNA, if you are talking about the sequence, it is the ATGC. DNA contain your, it's nothing but nucleotide, adenine, guanine, cytosine, thymine, ATGC. If you are talking about RNA, so in place of thymine, it is uracil. So this sequence of the DNA, when the structure we know about the DNA, all the organism which is present in this world need to be sequenced. Means we need to know their genetic structure sequence. Excuse me. Can I stop it for 1 minute?
Okay, sir. Okay, sir.
Just 1 minute, okay?
Okay.
I'm so sorry. Actually, our dean sir was called for important things to discuss. So I just sorry I was interrupted in between.
It's okay, sir. It's okay.
Okay. So I was talking about the 21st century sequencing, next generation sequencing. So human genome sequencing has been already done. Here the sequencing means your all the genetic materials of the particular organism has been sequenced. Sequence means what is the structure of their genetic material, the ATGC sequence. Okay, so now I will show you the, I will take you to the another in the website. Just hold on. Okay, I will. So if somebody is interested in the bioinformatics kind of the work, they have to go to this website NCBI. Is it my slide? This website is visible? Dhansa sir?
Yes, sir. It's visible.
It's visible, na?
Yes, sir. Yes, sir.
Okay. So this National Center for Biotechnology Information, this is the website, this is the database. Now, there is 3 type of the database used to be there: primary, secondary, and tertiary. This NCBI is the primary data set, which contain your all the information related with particular specific organism which has been sequenced. In this website, there is other things, are many things are there. So you just need to go in this National Center for Biotechnology Information. You have, so I already opened some tab here. So this is how it looks like, your NCBI, National Center for Biotechnological Information website. If you see here, I will explain some of the things on this. Many things I also learning now. Okay. So here, if you see here, this all database, it contain your all the different things: assembly, biocollection, bioproject, biosample, books, clean variety, then DB variety, gene, genome. Our work is here in genome. Okay. So in genome also, and also here the transcriptome also data you can get some of the things. Your proteomics also some of the things are here. Not completely, because the here in primary database, everything whatever is present, it is like a reference sequence. Means reference sequence in the sense of particular researcher working on to sequence some organism, and they got some sequence, so they will submit that sequence to NCBI. That sequence is the raw sequence which they have done the research. So this is the your raw data which NCBI is collecting and keeping all in their format, specific format. The secondary database is the your a much more of quality data. Quality data in the sense of the researcher is taking that raw sequence, and they will do the specifically, for example, like your suppose one specific signaling pathway is there which is responsible for that, so they will give that only that information only. Okay, that is the secondary database. Third database is the more specific. Okay, so here in this website you can go and scroll it and see that all the what are the things are there. It is quite good for MESH. This is the your search keywords actually. NCBI website, NLM catalog, nucleotide specific sequence, you can get it here for any organism. TMC, protein cluster, your protein family models, SNP, single nucleotide polymorphism, taxonomy toolkit. So I have opened already another tab, because sometime what will happen, this website is accessed by in whole world. So while adding that, sometime it is slow. So I just keep open the tab, so it will save the time. So I have given the keyword bacillus. Okay, this is the your it having a this bacillus having a your prokaryotic as well as the eukaryotic also. This walking stick is the your genus, bacillus is the your plant kind of thing, which is walking stick, and this bacillus is your prokaryotics. Okay, now here if you see that result found in 32 database, I just write it, and bacillus I open this one separately previous itself. So what are the thing, क्या चीज इसमें दिया हुआ है, बुक सेल्फ, तो इस वर्ड से रिलेटेड इस वर्ड से रिलेटेड बुक सेल्फ कितने हैं, MESH वर्ड कितना है, असेंबली, बायो कलेक्शन, बायो प्रोजेक्ट, बायो सैंपल. यहां पर हम देखना चाहेंगे जीनोम. ओके, अगर आपको न्यूक्लियोटाइड देखना है आप न्यूक्लियोटाइड देख सकते हैं, टेक्सोनॉमी के बारे में देखना है तो टेक्सोनॉमी देख सकते हैं, क्लिनिकल ट्रायल्स में कैसा क्या क्या चल रहा है ये सब चीजें देखी जा सकती है, जीन्स, सो ये सब चीजें, प्रोटीन्स कौन कौन से हैं उसमें, कंजर्व डोमेन है या प्रोटीन्स, पबकेम, इसमें all your articles. Okay, so this, when we click it in the bacillus, another, if we see that search in the bacillus, these are the different results are there for that organism. These are the different organisms which is present. Okay, so Bacillus cereus, Bacillus thuringiensis, Bacillus subtilis, Bacillus anthracis, Bacillus anthropus, Bacillus amylocephasians. So according to your wish, where you want to work, in what kind of the organism, either it is the plant or it is the animal or any bacteria, you can just need to search and it will show. So I have shown that in the next step. When I clicked it here, it will open and take you here. So here, if you see, this is your summary of your all the sequence, your genome assembly 716 sequence read length, then all the information, publications, representative reference genomes. This is a dendrogram of that. Okay, for more detail you need to go through one by one. Okay, so here important thing is what, here we need to check the genome sequence. Okay, so here, okay, in the, this is the your genome sequence of that Bacillus thuringiensis. When we'll click it out, this sequence will be, so when you see here FASTA, F-A-S-T, FASTA format. FASTA is the format of your, which is, I will show you in the next step. First we will see that here. This all is the accession number, reference sequence, your ATCC. This is the specific of that organism, different strain. How many genes are there total, CDS genes, coding, config. This is after doing the sequencing and all this, all the details are here. Now, here also you can download the FASTA sequence send to. You can take that the file. When you see the file, it can be format, different format used to be there. GenBank, FASTA, GenBank full, XML. These are the different formats of the same sequence of the FASTA format. So we generally, when we do that, suppose for example, there are so many organisms already available which has been already sequenced. Suppose some person is doing some research. Okay, he got some new strain which is not existing. Okay, so how to know that one? So what that new strain, they have to sequence. That new strain will be sequenced and that sequence need to be compared with the existing sequence. So to compare that, we require this FASTA format. This is your FASTA format. When you see the FASTA format, we can download also this one. This will be some few MBs. I've directly opened here. If you see that here there is a minimum in the FASTA format, there is always 2 lines is minimum line will be there. Okay, and it is always start with this symbol, greater than symbol. You can see this greater than symbol in the starting, and this is the heading of that. So what is this? Bacillus thuringiensis 0/. This is your genome sequence, whole genome sequencing, shotgun sequencing. And you can see here, this is your sequence exactly. So you can see TTA, this is thymine, adenine. So this is the sequence of your whole of this Bacillus thuringiensis. Okay. Now, so if suppose I am doing some research, I got some new organism which has having a bacillus. That is the unique. So what I need to do, I will sequence that one by wet lab. That sequence I will send it to for their ATGC to know that, and I need to compare. We need to do the BLAST, basic local alignment sequence tool. Okay. So this BLAST, what it will do, it will compare what organism is present. It will check what are the similarities there. Means, for example, this suppose in my unique organism contain this much sequence, and there is some change in the next third line, and other is the same. So that will be, we can know by the phylogenetic tree. Okay. So this is how it is revolutionized in the 21st century, is that next generation sequencing, we are using all kind of your sequencing of your organism, and we are keeping the databases. Now here in this databases is the important tool to target or change in this. Okay. So that is for your related with the bioinformatics in the next generation sequencing. This is just a tip of iceberg. Okay, there is so many things are there in this. I am also new in this. I am also learning, but I have to take as a challenge to give to ignite you all to go in this field, because there is a two type of the research used to go. Okay, let me take to another, just a minute. Okay. Okay. So how it is like in new generation sequencing, the water organism which is present, they all are getting sequenced now to know their structure, which is not available, and they will check the target of it. Okay. So this is just a glimpse of that. So that was the research point of view. There is another next slide. This is another area. This slide is visible, is it?
Yes, it is visible.
Yeah. So this I have shown that just a one target kind of thing, how it is in type 1 disease, type 1 diabetic, what will happen? Your beta cell will get destructed because of autoimmune disease. Okay. So I'm not going to go for detail of this. This I'm just showing up as one kind of generally inhibit this DPP4. This is the DPP4 enzyme. What it will do, generally it will stop this GLP, glucagon-like peptide, which is responsible for your insulin, means to decrease the diabetic glucose level, sorry. And another is that this antibody we are using for targeting specifically to stop this process of your destruction of the beta cell. Okay. So now I'll finish my talk here. So I will give 15 minutes for discussion. If anyone having any query.
Okay, thank you, sir. Students, अगर आपकी कोई क्वेरीज हैं, टीचर्स, रिसर्च स्कॉलर्स, तो आप पूछ सकते हैं. सर, मैं एक पूछना चाहूँगा सबसे पहले. हेलो.
यस सर, सुन रहा हूँ.
अभी रिसेंटली कुछ पेपर्स आए हैं, एंटी कैंसर रिसर्च पे ही है. तो टर्म्स यूज हुआ है PD1. जिसको टारगेट किया जा रहा है बेसिकली कैंसर के ट्रीटमेंट के लिए। तो इस बारे में अगर कुछ आप इस बारे में आप कुछ बता पाएँ तो।
What is the full form? I think it is signaling molecule, if I am not wrong.
PDA, I think it's related a protein drug.
No, I don't have the directly idea on this, sir.
Okay, sir. Okay.
Sorry.
ओके सर। सर एक और डाउट था मेरा, जैसे कुछ टेक्निक्स हैं क्या जिसके थ्रू हम फर्मेंटेशन को डिले कर सकें?
फर्मेंटेशन को डिले करने के लिए रिप्रेसर यूज किया जा सकता है। मतलब आपका जैसे फर्मेंटेशन प्रोसेस में क्या होता है, आपका जो सेल्स है वो डिवाइड हो रही है। यहाँ पे दो टाइप की चीजें हैं, आप किस चीज पे फोकस कर रहे हैं। मतलब आप प्रोडक्ट चाहिए, मतलब सिंथेसाइज्ड प्रोडक्ट चाहिए, या फिर द सेल्स ही प्रोडक्ट है।
एक्चुअली सर ये नेचुरल लिक्विड प्रोडक्ट है, जो लंबे समय तक या 3 या 4 घंटे से ज़्यादा अगर हम इसको स्टोर करके रखते हैं तो उनमें ऑटो फर्मेंटेशन हो जाता है और वो यूजलेस हो जाते हैं। तो हमको लंबे समय तक प्रिजर्व करके उनको रखना है, तो कैसे हम फर्मेंटेशन को डिले कर सकते हैं?
फर्मेंटेशन का यहाँ पे प्रॉब्लम किस वजह से, ये दूसरे इन्फेक्शन में से दूसरे माइक्रो ऑर्गेनिज्म के ग्रोथ की वजह से हो रहा है।
हाँ सर, हाँ, नेचुरली
तो प्रिजर्वेटिव
एनवायरनमेंट में रखने पे ऐसा हो रहा है उसके साथ।
सो एक चीज तो देखिए, मैं इसको अब इसमें थोड़ा मैं आपको कुछ हिंट कर सकता हूँ, डायरेक्टली तो नहीं होगा। जैसे आपका वो ऑर्गेनिज्म है, उसका इन्वायरमेंट क्या है, कि वो किस इन्वायरमेंट में स्पेसिफिक टेम्परेचर और प्रेशर में उसका है, और उसमें जो कंटामिनेटिंग माइक्रो ऑर्गेनिज्म है जो उस टेम्परेचर में ग्रोथ कर रहे हैं, तो उसे आप स्टॉप करने के लिए कर सकते हैं, पहला चीज। दूसरा क्या है कि मुझे जहां तक मेरा ख्याल है, आप जो बोल रहे हैं फर्मेंटेशन के लिए, उसकी सेल्फ लाइफ मेरे ख्याल से 3 से 4 दिन ही है क्या?
3 से 4 घंटे ही है सर, उसके बाद वो उसको यूज नहीं कर पाते।
तो इसमें सबसे अच्छा मैं ये कहना चाहूंगा, फर्स्ट तो उसको देखना पड़ेगा कि उसकी स्टेबिलिटी किस में है, क्योंकि जनरली आपका कोल्ड इन्वायरमेंट में रखने से उसको लॉन्ग टाइम पे स्टोर किया जा सकता है, लेकिन उसको ऑप्टिमाइज करना पड़ेगा। ये जनरल टर्म है, आप मुझे वेरी स्पेसिफिक बताएंगे तो उसके थ्रू से मैं कुछ बता पाऊंगा।
एक्चुअली सर छत्तीसगढ़ में एक सल्फी करके नेचुरल लिक्विड प्रोडक्ट आता है बस्तर एरिया में। सल्फी तो उसको हम लंबे समय तक प्रिजर्व नहीं कर सकते,
तो
3 घंटे बाद भी टॉक्सिक हो जाता है।
सल्फी है वो आपका डेयरी प्रोडक्ट है, या मतलब मेन क्या है, ट्रेड नेम है, सल्फी क्या है?
नहीं, ट्री प्लांट प्रोडक्ट है, लिक्विड जूस है वो।
लिक्विड जूस है,
हाँ।
ओके, आई नीड टू गो थ्रू, मेरे को देखना पड़ेगा, उसके बाद ही मैं बता पाऊंगा।
ओके। अदर स्टूडेंट्स, आप लोगों को कुछ डाउट्स होंगे तो?
गुड आफ्टरनून सर, मैं डॉक्टर कविता कृष्णमूर्ति। मैं कुछ बेसिक क्वेश्चंस आपसे करना चाहती हूँ रिगार्डिंग बायो इन्फॉर्मेटिक्स। सर, बायो इन्फॉर्मेटिक्स में बहुत सारी इंफॉर्मेशन बहुत सारी डेटाबेसेस में सेव हैं, तो बिना लैब, जो हमारा ड्राई लैब का कॉन्सेप्ट है, किस तरह से हम इन डेटाबेस का यूज करके फर्दर हम रिसर्च में आगे बढ़ सकते हैं, सिर्फ डेटाबेस का यूज करके?
डेटाबेस में यूज करके आप ऐसा कर सकते हैं, देखिए इसमें जैसे आपका, हालांकि मैंने पहले ही बताया कि मैं इसको सीख रहा हूं करके, लेकिन मैं आपको इसको कुछ आइडिया दे सकता हूं। जैसे आपका प्राइमरी डाटाबेस है, तो इसमें बहुत सारी चीजें हैं जो अभी भी उसमें हिडन कुछ चीजें हैं, हिडन इन द सेंस ऑफ सेकेंडरी डाटाबेस बनाने के लिए आप एक ही ऑर्गेनिज्म के डिफरेंट सीक्वेंस को कंपेयर करके उसके बारे में आउटपुट निकाल सकते हैं। जैसे आपको टारगेट डिफरेंट पॉइंट है, जैसे मैं आपको थोड़ा सा बताना चाहूंगा इसमें। जैसे वेट लैब में हम कोई काम करते हैं, आपके ड्राई लैब में मैं आऊंगा, पहले मैं आपको बताना चाहूंगा। जैसे वेट लैब में हम कोई काम कर रहे हैं और उसके सिग्नलिंग पाथवे हमें पता लगी कि पर्टिकुलर सीक्वेंस में हमें टारगेट करना है, ठीक है। तो हर एक ऑर्गेनिज्म का एक स्पेसिफिक सीक्वेंस होता है। तो वो जो सीक्वेंस में आप अगर किसी चीज को टारगेट करना चाहते हैं, जैसे आपका अमीनो एसिड में जो उनके प्रोटीन रिस्पॉन्सिबल हैं डिजीज कॉज के लिए या फिर और कोई प्रॉब्लम क्रिएट करते हैं, तो उस टारगेट को अगर उस प्रोटीन की सिंथेसिस ही हम ना बनाए या फिर उसको फाल्टी कर दे, फाल्टी इन द सेंस ऑफ सपोज ए सीक्वेंस है, एटीजीसी का सीक्वेंस है जो ट्रांसक्राइब हो रहा है और एक प्रोटीन बन रहा है, ठीक है, और उस सीक्वेंस में अगर हम चेंज कर दिए कुछ, अगर एडेनिन की जगह हम थायमिन कर दे, तो जो प्रोटीन हमारा जो बन रहा है वो क्या है, वो फाल्टी प्रोटीन है। वो फाल्टी प्रोटीन है तो उसकी एक्टिविटी नहीं रहेगी। ये जस्ट एक एग्जांपल है आपका उस प्रोटीन सीक्वेंस में चेंज करने का। इसे आप डीएनए में चेंज करके देख सकते हैं, न्यूक्लियोटाइड में चेंज करके देख सकते हैं, आपका प्रोटीन सीक्वेंस में चेंज करके देख सकते हैं, और दूसरे जितने भी डाटाबेसेस है उसके कंपेयर करके आप उसका आउटपुट निकाल सकते हैं।
Okay, thank you, sir, for giving this idea. Thank you.
Any other queries from students, teachers? Okay. Okay, now I am inviting Mr. Virendra Sahu sir for vote of thanks.
Thank you, sir. Good afternoon, everyone. To the one and all present here, respected Dr. Hanuman C. Rathore sir, and all the participants. It is such an honor for me to get the opportunity to thank you all on behalf of the Chemistry, Zoology, and Botany Department, Government Lahari PG College, Chirmeri. I extend my gratitude to our keynote speaker, Dr. Hanuman C. Rathore sir, to take out time from his busy schedule to grace the event and for his valuable talk on such an important topic, biotechnology research in 21st century. Thank you for inspiring and encouraging us with your words on this special day. I thank to Principal or Government Lahari PG College, Chirmeri, for providing immense support to make the National E-Workshop successful. I must thank the organizing team. Thank you everyone once again for making it a great success. Thank you, sir.
थैंक यू
Thank you, sir.
धनसाय सर, मैं थोड़ा कुछ बोलना चाहूँगा।
बिल्कुल सर, स्वागत।
अभी टाइम वैसे 8 मिनट बचा हुआ है, तो स्टूडेंट को कुछ कहना चाहूँगा मैं। हालांकि वोट ऑफ थैंक्स हो गया, लेकिन फिर भी मैं अपनी तरफ से कहना चाहूँगा विद द परमिशन ऑफ द चेयर। पहले तो मैं आप सभी को धन्यवाद देता हूँ। मैं एक चीज़ बताना चाहूँगा कि सभी स्टूडेंट लोग के लिए कि आपको अगर कांटेक्ट करना है तो मेरा ईमेल आईडी, फ़ोन नंबर दिया हुआ है, आप कभी भी कांटेक्ट कर सकते हैं। इसके बाद मैं ये स्लाइड शेयरिंग हटा रहा हूँ, उसके बाद फिर मैं थोड़ी चर्चा करूँगा आप सभी स्टूडेंट लोगों से। मेरे ख्याल से 60 पार्टिसिपेंट्स हैं यहाँ पर।
हाँ सर।
Yes, sir.
मैं सभी स्टूडेंट को ये कहना चाहूँगा, मैं भी हिंदी मीडियम में पढ़ा हूँ, और आप लोग को ये भी कहना चाहूँगा कि धनसाय और मैं, हम लोग बैचमेट हैं। अब धनसाय सर ने बताया है नहीं बताया मुझे पता नहीं है, मतलब चेयर के परमिशन की तरफ से मैं थोड़ा सा चर्चा कर रहा हूँ, इनफॉर्मल टॉक है, आप लोग माइंड मत करिएगा। अगर प्रिंसिपल मैडम है यहाँ पर या फिर और भी प्रोफेसर्स, ठीक है।
नो, इट्स ओके सर, कंटिन्यू।
तो धनसाय सर ने बताया है नहीं बताया मुझे पता नहीं है, लेकिन धनसाय और हम दोनों बैचमेट हैं, और मैं भी छत्तीसगढ़ का ही रहने वाला हूँ, और हम उस स्थिति को अच्छी तरह से जानते हैं और देखे हैं। तो मैं आप लोग को ये कहना चाहूँगा कि अगर किसी का इंग्लिश कमज़ोर है या वो इंग्लिश नहीं जानता है या कम है, तो भी आप ये ना सोचिए कि आप बढ़ नहीं सकते। मैं एग्जाम्पल हूँ आप लोगों के सामने।
Yes.
मैं हिंदी मीडियम में पढ़ा हूँ, और धनसाय मेरे बारे में अच्छी तरह से जानते हैं, और मैं ये भी कहना चाहूँगा कि मैं अपने नॉलेज के बस दम पे मैंने अपनी एमटेक की पढ़ाई की, मैं पीएचडी भी किया और अभी मैं सेंट्रल यूनिवर्सिटी में काम कर रहा हूँ। तो ये मत सोचिए कि ये नेगेटिव थॉट आप अपने दिल से निकाल दीजिए कि आप कुछ नहीं कर सकते। ये आप लोग की इंकरेज करने के लिए है, और ज़्यादा समय नहीं हमारे पास, लेकिन फिर भी आप लोग कोई भी चर्चा करना चाहें, चाहे स्टडी से रिलेटेड हो या और भी कोई बातें हैं, तो आप चर्चा कर सकते हैं।
यस
ठीक है। और बल्कि मैं तो सर से ये कहना चाह रहा था, ये वो मतलब बहुत ज़्यादा इंटरेक्टिव रहता, ज़्यादा अच्छा रहता। मैं अपनी तरफ से ही बोला जा रहा हूँ। अगर ये
नहीं, आज हम सुनने को ही प्रेजेंट है सर सब यहाँ, और बहुत अच्छा स्पीच डिलीवर था आपका, बहुत सारी चीज़ें जानने को मिली अभी, क्योंकि प्रेजेंट में जीनोमिक्स बहुत ज़्यादा चल रहा है, जीनोम सीक्वेंसिंग, उसमें बहुत अच्छे आपने लेक्चर डिलीवर किया। सर ये जीनोमिक्स में अपना ये सिकल सेल एनीमिया के बारे में रिसर्च हो ही रहा है ना सर अभी?
सिकल सेल एनीमिया में भी रिसर्च हो रहा है। ये देखिए, ऐसा है कि कुछ कारण जो है, जैसे हम लोग भी ना, रिसर्च इतना वास्ट फील्ड है, मैं बहुत कम जानता हूँ, मैं भी अभी सीख रहा हूँ किसी भी फील्ड में। मैं जहाँ पे हूँ भी, यहाँ पे मैं अभी सीख ही रहा हूँ, थोड़ा बहुत ही जानता हूँ, ज्यादा नहीं। ये मेरा सोचना है, क्योंकि
Yes.
जब आप सोचेंगे कि आप किसी चीज के बारे में कम जानते हैं, तब और ज्यादा काम करें। सिकल सेल एनीमिया के बारे में आपको चेक करना है, तो उसके बारे में आपका जीनोम सीक्वेंस, उसके जो ऑर्गेनिज्म है जो रिस्पॉन्सिबल हैं,